Hi everyone, I'm going to present on the use of mitochondrial DNA in phylogeography and paleoecology. First, I'll just go over some concepts. So phylogeography can be described as the study of historical processes that have resulted in present biogeographic distributions of populations and individuals. Paleoecology studies the interactions between organisms and their environment across geologic timescales. And a quick review for mitochondrial DNA. This DNA is passed on maternally, and it's not involved in recombinant processes, and any variants found are due to mutations. These mutations form groups of haplotypes called haplogroups. These groups tend to be geographically restricted and can distinguish different populations. To see how these all come together, I found a video by National Geographic that explains it in simple terms. We won't watch the whole thing, but it should be about a minute and a half or 2 minutes.
So what's the science we use to study the ancient migratory paths of your ancestors? It's something called population genetics, and it helps us to piece together how humans populated the Earth. Here's how it works. Historically, people close to each other tended to share more of those genetic markers, the little DNA typos passed on from parents to their kids, than they did with people on the other side of the world. Makes sense. After all, who were they more likely to meet, fall in love with, have babies with? The people living nearby. Over time, this created regional patterns of genetic variation. So the people living in, say, northern Spain had more markers in common with each other than they did with people in Indonesia. These regional patterns of variation allow us to track the migratory paths of your ancestors. By looking at where in the world your markers are found, we can figure out where you came from. And by looking at the order in which these markers have occurred over time, and where in the world they originated, we can actually track the journey your ancestors followed from an African homeland to where you came from more recently. How do we find these markers in the billions of A's, C's, G's, and T's? It's not easy. It's a little like finding needles in a haystack. That's because when a mom and a dad have a baby, their chromosomes are copied and passed on to their child, but not in a straightforward way. Instead, mom and dad's chromosomes are shuffled and they're passed on to their kids. Children. It's called mitochondrial DNA, or mtDNA for short. It's much smaller than the DNA that makes up our 23 pairs of chromosomes. But it contains the same kind of markers, the little genetic typos that pass unshuffled from generation to generation on the maternal line. So by looking at markers on the Y chromosome and mitochondrial DNA, we can connect people alive today to ancestors that lived tens of thousands of years ago. Over the last several years, the Genographic Project has...
So now that we've gotten some of the main concepts out of the way, I can dig into the articles that I found. The research paper by Ludovic and Cooper, Using Ancient DNA to Understand Evolutionary and Ecological Processes, is a paper that presents the use of ancient mitochondrial DNA in different studies, and it broadly explains the many uses for this type of proxy. And then the next two papers then present specific studies undergone by the authors themselves, and it reflects the uses discussed in the first article. So this paper was quite extensive and covered a lot of important topics within the use of mitochondrial DNA, but I'm just going to discuss the more important or interesting key points. First, the paper describes why researchers use mitochondrial DNA. For one, it helps us understand population processes and dynamics, particularly with respect to the environmental and climate change impacts. This information helps us understand genetic diversity in modern populations. It is also used to interpret fossil records and evolutionary timescales. The next key point they discuss is the accuracy of the fossil record. Using DNA has uncovered common mistakes done through morphological studies, which is taxonomic oversplitting. This means that one species can be identified as many different species. For example, the giant red-tailed moa from New Zealand. There were many proposed different Dinornis species, which is a type of moa, And they were actually the same species with sexual dimorphism. So 1 and 4 here are two different types of Dinornis species. And originally, number 4 was thought to be many different types of species. The females of this species can be 150% taller than the males, so it's easy to see why they'd separate them into different species. Bison DNA, while having had 52 species or subspecies proposed, show two lineages from Bison priscus, giving rise to the two living species, Bison bison and Bison athabascae. It is suggested that many paleontological studies could have underestimated the temporal spatial variation shown by many mammals. Researchers also use ancient mitochondrial DNA to measure population dynamics and explore the past environmental changes on biological communities. The bison is again used as an example. Studies have shown that there was a demographic collapse of bison in Beringia long before the arrival of humans. Similar to the previous point, population genetics over time is studied. There is a common theme with the loss of genetic diversity prior to the last glacial maximum in mammals, such as the brown bear, Arctic fox, bison, and lemmings. This contradicts previous studies which suggested a reduced genetic diversity due to the glaciers. So the loss of genetic diversity seen before the last glacial maximum is likely to have occurred because of the high frequency of rapid climatic oscillations during the late Pleistocene, which began 129 years ago with the Eemian interglacial period, which was 1 to 2 degrees warmer than the Holocene. So this interglacial period is believed to have greater seasonal temperature variations than today and caused a lot of the loss of genetic diversity. Another example of research on population genetics over time includes endemic birds of Hawaii. Just as it was believed that the last glacial maximum caused loss of genetic diversity, it was believed that humans caused the reduction of genetic diversity in many Hawaiian birds. Ancient DNA suggests that the genetic diversity of petrels, which is on the left there, remained high during human colonization, likely due to the long generation time and lifespan. The Hawaiian goose on the right, however, did suffer genetic loss from hunting and habitat loss from humans. So they responded differently to the arrival of humans on a genetic level. Environmental change and its impacts is extensively studied, and mitochondrial DNA is an extremely useful proxy. Many plants and animals were revealed through DNA in sediment cores, even without macrofossils. Siberia cores show different plants through stratigraphic layers, showing the shift from herbs to shrubs over the past 300,000 years. New Zealand coastal sediment cores revealed now extinct megafauna. Ancient boreal tree DNA was discovered in sediment cores of the Norwegian island Andoya, in the horizons dated back from 19 to 22,000 years ago, a time when ice sheets were thought to completely cover Scandinavia. This suggests refugia during the last glacial maximum, possibly from organisms surviving on nunateks. Furthermore, studies from DNA, pollen, and macrofossil proxies show poor overlap, and more accurate reconstructions are done when using the proxies in combination. Extinctions are studied using DNA as well. However, mitochondrial DNA is not always the most helpful. For a full reconstruction on a species' history, using past species and its descendants is best. So if they've gone extinct, there is nothing to compare the DNA to. I'm going to briefly sum up the important points from this article. First, mitochondrial DNA helps us interpret fossil records. It helps researchers reconstruct population dynamics in response to environmental change. It allows us to study genetic diversity and understand environmental changes through geological time. Lastly, a combination of ancient mitochondrial DNA, present mitochondrial DNA, and other proxies allows for the most accurate information. This next paper I found really interesting, titled Glacial Survival or Late Glacial Colonization, Phylogeography of the Root Bowl in Northwest Norway. It directly relates to the first article, in that it discusses the possible survival of organisms on Andøya Island during the last glacial maximum. This study suggests that northeast coastal islands of Norway had permanent ice-free conditions, which the root bowl and other organisms survived. Its aim was to understand the origin of European populations after the glacial period. It also refers to the controversial nenetic hypothesis and in situ survival hypothesis. The nenetic and in situ survival hypotheses reflect this study's hypothesis that some biota survived mostly glaciated regions on land that remained ice-free, such as nenetics. This is a nenetic here. It's the areas of land that are higher than the ice sheet, so they're not completely covered by ice, and that's where they are thinking that some of these species survive. For the methods, they tested DNA extracted from ethanol-preserved root bowl tissue. Mitochondrial DNA was analyzed from 46 root bowls from 19 localities. The mitochondrial cytochrome B gene was amplified using the polymerase chain reaction. Statistical analysis was done on nucleotide diversity, haplotype diversity, and DNA divergence. They then did phylogenetic analysis using different types of algorithms. The phylogenetic analysis is quite complicated, but this figure can help visualize it. So you can notice two distinct groups. You have the ones in yellow and then the ones in red, and they kind of split from where the arrows are showing here. And if you notice, so this has Andøya, Rinoya, and Ringovacea. And then if we look on the next figure, they're not actually that close together. So it was with the help of mitochondrial DNA that they discovered the relation. They found two different phylogroups and designated them Andøya and Phenoscandian, which was seen on this slide. So the yellow is the Phenoscandian and the red is the Andøya. The Andøya sublineage phylogroup was from the islands of Andøya, Ringovacea, and Rinoy, labeled in red on the right map, as well as western areas of Russia were included. The Phenoscandian phylogroup was from 3 Norwegian islands of Kvalia, Hekoya, and Arnoya, marked in blue. And areas of northern Sweden and Finland were included in that group as well. They concluded that the Phenoscandian phylogroup recolonized through Finland, as other studies have already suggested. So on this left map here, they figured that the Phenoscandian group just kind of came in from here through Finland. However, the Andøya group, which carried a unique cytochrome B haplotype, exhibited a specific evolutionary branch. Along with plant microfossils, this information indicates a bowl glacial survival in situ. This presents the distinct possibility that there was an uninterrupted ice-free period on the island during the last glacial maximum. The high mitochondrial diversity of today's bowls indicate the root bowl is capable of survival in small isolated populations, and there may have been small isolated populations surviving on neighboring islands. So the yellow area here marks the newly suggested area of refugia. And they figure that on top of this yellow area here, that's where they recolonized from, versus the pink area here, which was originally suggested. For the last article, I'm just going to quickly touch on a couple of the key points. They relate to the first article and explain the importance of studying mitochondrial DNA for human history. As briefly mentioned before, mitochondrial has a high evolutionary importance. It is inherited from mothers and can be traced through many generations. The mutations from mitochondrial DNA form haplogroups which tend to be geographically distributed, allowing for researchers to genetically distinguish populations. This article summarizes it perfectly, stating, ancient mitochondrial DNA retrieved from museum specimens, archaeological finds, and fossil remains can provide direct evidence for population origins and migration processes. They also discuss the mitochondrial Eve, and she lived in the sub-Saharan Africa 200,000 years ago. She was the last female common ancestor. They discovered that migration processes out of sub-Saharan Africa, and it's proven by mitochondrial DNA dated back 85,000 years ago, 27,000 years ago to 16,000 years ago. The last glacial maximum made Europe uninhabitable. And all these things can be proven through mitochondrial DNA. Some of the key points that, just to wrap up real quick, each article brings up the question of survival during last glacial maximum, and they suggest different areas that were actually ice-free, previously thought to be covered with ice sheets. Each article also states how useful mitochondrial DNA is as a tool in studying population dynamics and environmental change. They also suggest that using ancient and present mitochondrial DNA alongside other proxies for the best accurate reconstruction of the past. Lastly, here are some references that I use just to clarify different topics. They're really interesting reads, and they give lots of detail on the different types of genetic variations in different species, and it just gives you a better understanding of the topic in general. Thanks, everyone.